Hello everyone, this is video number 7 for the immune system, and in this video I'm going to talk about activation of helper T cells and what do they do after they're activated, and what is their effector function in an immune response. Before I do that though, just a reminder, for this chapter I made a very detailed outline of all the topics that we're going over, right? So it includes maturation, activation, and the immune response in general. And you could use it, you might want to use it as a companion as you're going through the videos. Also, another reminder, there are worksheets posted to this module. Some of them are blank, some of them are filled in. So the steps in maturation and the steps that occur upon activation and how activation occurs. All right, so just a reminder for you. And in this video, just to take a look at what we're going to cover. So steps 5, 6, 7, and 8 here in the life of a lymphocyte. And we're going to apply these specifically to the helper T cell. And so that includes activation, clonal expansion, the effector response, what does it do in an immune response, and memory cells. This slide is just a reminder what an immunocompetent helper T cell looks like. So it is equipped with a specific antigen receptor, the TCR, and also helper T cells have a CD4 co-receptor. So all of these are meant to represent receptor and co-receptor. They're paired, and there are 100,000 or so of these per immunocompetent helper T cell. Okay. During activation, what's going to happen is the T cell receptor is going to bind to a foreign antigen, and helper T cells are extremely picky about how this antigen is presented to them. It can't be any old form. It can't be a virus particle. It can't be a bacterium. It can't even be an antigen on the surface of a body cell. It must be presented only by an antigen presenting cell. Okay? And the form that the antigen presenting cell must present it in is coupled to MHC2. Okay? So unlike B cells, which the antigen receptor on their surface can bind to an antigen in any form, for helper T cells, extremely picky, only can be activated by an antigen presented by an antigen presenting cell, such as a dendritic cell. The co-receptor on the surface of the helper T cell is CD4, and this is going to be involved in binding to MHC2. So it's kind of like a lock and key, right? MHC2 is found only on antigen-presenting cells, and that interacts with CD4, which is found on the surface of helper T cells. Okay. Just to review a little bit about antigen-presenting cells, I talked about these earlier, but since we're going to be talking about them in this video, just to remind you, these are key cells, and they are phagocytic. So if they internalize some kind of pathogen or foreign antigen, they're going to degrade it, and they're going to couple pieces of that antigen, the antigenic determinants, couple it with MHC2 and put it on their surface. And that combination, MHC2 with a foreign antigen, is what's going to be evaluated and possibly bound by immunocompetent helper T cells. The major types of antigen-presenting cells are dendritic cells, macrophages also, and B cells can act as antigen-presenting cells. The dendritic cells are key antigen-presenting cells because they're involved in the initial activation of helper T cells. These dendritic cells sit in the skin and also in lymphatic organs and in the mucous membranes. And when they take something in and phagocytize it and then break it down and put it on their surface, they migrate away from that position to a location where T cells are located, so that increases the chances of exposure of a helper T cell. And this is just a figure of a dendritic cell. So it has all these spikes. Those are dendrites. That's how they got the name. And also that increases the chance of exposure, number one, to a pathogen or a foreign antigen, so it can be taken in, and also exposure to helper T cells. All right, so let's focus in on specifically what happens in the activation of a helper T cell. I prepared a short video to describe that, so let's do that now, and we'll come back and review that in the PowerPoint. All right, so let's talk about lymphocyte activation for T cells now. For B cells, I forgot to mention this on the previous video, but for B cells, the antigen can be in any form. It can be a bacterium, it can be a virus, it can be a molecule dissolved in the lymph or in the blood plasma. It can be an abnormal body cell. Any form, as long as it binds to the BCR, B cell receptor. For T cells, they're very picky about the form that the antigen is in order for them to be activated. So for a T helper cell, starting with the helper cells, T helper cells are equipped with a T cell receptor, shown in blue here, and a co-receptor. T helper cells have the CD4 co-receptor. So it's two things involved in binding. And the only form that an antigen can be presented in that a T helper cell will recognize is if it's presented by an antigen presenting cell. So here's an antigen presenting cell. This could be a dendritic cell. I've drawn it like a dendritic cell, but there are others as well. Macrophages, for example. An antigen presenting cell does this. It's phagocytic. It takes in some foreign substance, say a bacterium. It breaks it down, and then it couples pieces of that foreign antigen to MHC2 self-antigens, and then it puts those on the surface. So here is MHC2 in black, and these are coupled to different parts of the bacterium that it broke down. So let's see along comes a T helper cell, and it has a T cell receptor that recognizes one of these antigens coupled with MHC2. So what happens is the T cell receptor binds to the antigen. It's an antigen receptor. And the CD4 co-receptor binds to the MHC2. You can think about it as a key fitting into a lock, right? If both of them fit by complementary structure, has to be both of them, not just one of them. If both of them fit, then you've unlocked the door, the lock, or whatever you want to call it, okay? So if there's complementary fit between these, this cell is going to be activated. If there's no fit, maybe they bump into each other, they don't match, this T cell will wander away, and maybe later another T cell that has an exact fit will encounter it, okay? But it has to be presented, the antigen has to be presented along with MHC2. A helper T cell will not be activated by a bacterium or a virus particle. It has to be presented by an antigen presenting cell in this form. And remember, only antigen presenting cells have the MHC2, okay? So helper T cells are very picky how the antigen is presented. What happens when it's activated? So helper T cell activation is triggered by binding to the foreign antigen presented with MHC2 by an antigen presenting cell. What happens next is going to be similar to what happens in a B cell activation. Clonal expansion. This helper T cell now is going to divide very rapidly, and it's going to form an army of helper T cells that have the exact same T cell receptor coupled with CD4 co-receptor. Now you have thousands of these cells. All right, most of these cells are going to become effector cells. For helper T cells, effector cells become secretory factories. They secrete lots of cytokine molecules. cytokines. Cytokines are signaling molecules, and the effect of the cytokines is these actually stimulate this cell, autostimulation it's called, to undergo cell division. So that's part of what happens in clonal expansion. But not only this T cell, remember this is a helper T cell, this is going to help other immune system cells to also divide. You're going to expand all of your immune system cells. So that includes B cells, it includes innate immune system cells like macrophages, natural killer cells, etc. Okay? That's what the T helper cell effectors do, secrete a soup of cytokines that has the general effect of increasing immune system cell division. Also, some of these clones of this T helper cell are going to be basically socked away in the body. They're going to travel through the bloodstream and the lymph, and they're going to take up residence in various lymphatic organs. And if you're exposed to this same pathogen later on, whatever it is, bacterium, then now you have an expanded number of these. And they're everywhere in the body, maybe just not in one limited location. All right, so that's T helper cell activation. Okay, so just to review, in helper T cell activation, there is actually two things that happen. So the first thing is there is binding between the T cell, the helper T cell, and the antigen presenting cell. Okay, and the binding is the TCR binding to the antigen, the foreign antigen, on the surface of the antigen presenting cell, and the CD4 co-receptor binding to MHC2. So like I said in the video, it's a lock and key. You must have both of those happening. Okay? So if that occurs and it's a stable binding, recognition is good, they will remain in contact for a long time, several hours at a time. Okay, and then after that activation, then the next steps are going to occur. So those are shown on this slide. The helper T cell is going to start secreting. This is part of the activation response. A cytokine called interleukin-2, IL-2. And this cytokine acts on the helper T cell itself to stimulate cell division. So this is part of the clonal expansion process. So clonal expansion occurs, and now instead of having just one helper T cell with that specific antigen, you now have thousands. Okay? So you've expanded the number of helper T cells specific to that foreign antigen. Of this expanded pool of clone cells, some of these are going to be effector cells. And the effector cells are the cells that make and secrete a whole host of different cytokines. These cytokines stimulate cell division of immune system cells in general. Some of them are going to act on B cells, and that's going to stimulate the humoral response to this specific antigen. B cells are also going to be able to recognize this foreign antigen. They're also going to stimulate cells of the innate immunity, so macrophages, natural killer cells, and other leukocytes. So the general effect of the helper T cell effector cells is to enhance the immune response in general. And the importance of that feature of helper T cell function is seen for diseases where helper T cells are impacted, their numbers are reduced. And we'll talk about some examples of those at the end of this chapter. The other cells in the expanded clonal pool of the helper T cell that was activated do not become effector cells. They do not secrete cytokines, but they are sort of stored away in the body and various lymphatic organs should you experience another encounter with this same foreign antigen. So those are the memory helper T cells. Memory cells are formed in the B cell response, in the helper T cell response, and you'll see in the next video also in the cytotoxic T cell response. Okay? So this slide is just summarizing the activation of the helper T cell. And so here is the antigen presenting cell presenting a foreign antigen coupled with MHC2. Here is a helper T cell that's responding by binding the antigen with its antigen receptor, and MHC2 is interacting with the CD4 co-receptor. Here is the response, secretion of IL-2, which auto-stimulates this cell. It undergoes clonal expansion, and then some of these are going to secrete other cytokines. Those are going to be the effector helper T cells. Others of them are going to just be stored away in the body. Those are the memory T cells. Okay, a little bit more about the effector response of helper T cells. So when a helper T cell is activated, it's going to leave the lymphatic organ and migrate to the site of an infection. And once there, it's going to secrete this cytokine soup, which is going to be helping to stimulate the activity of all these other cells, as I mentioned before. B cells, cytotoxic T cells, and cells of the innate immune response. So this slide is just illustrating, it's not a very good illustration to be honest with you, but this is the effector response of an activated helper T cell secreting a bunch of cytokines to stimulate the immune response in general. Okay, so I'm going to end this video here, and the next video will discuss activation and effector response of the cytotoxic T cells. See ya.